All right, so today we're going to talk about recombinant genes. So, what is a recombinant gene? A recombinant gene is a gene that has been artificially created by combining DNA from different sources, typically inserted into a host organism for expression. This is part of recombinant DNA technology, but this specifically relates to genetic engineering and the sequence itself. So, we can't do any of this without vectors, and vectors are nucleic acid molecules that deliver a gene into a cell. There's a couple of different useful properties for them. They are small enough to manipulate in a lab, and they can survive inside of cells. They contain recognizable genetic markers that ensure genetic expression of a gene. This can include anything from viral genomes to transposons and plasmids. So we can see here kind of the steps that go through to get a recombinant gene. So we've got a plasmid, and our restriction enzymes come in and cut the plasmid open. Then our DNA comes and attaches itself to that plasmid, the selected DNA, and ligase comes and forms the bonds. That plasmid is then inserted into the bacterium, and that bacterium is grown. So in this example, once that bacteria starts growing, it can start producing the human growth hormone, which we use for people that struggle to produce that hormone on their own. So we do need a couple of tools for DNA recombinant. We need restriction enzymes, DNA ligase, plasmid vectors, host cells like bacteria, yeast, and mammalian cells, and selectable markers. So things like antibiotic resistance, production of insulin, production of human growth hormones, so on and so forth. So there's a couple of different uses for recombinant genes, and we're going to kind of go through and look at each one of them. So, pharmaceutical and therapeutic applications. This is when protein synthesis, the first one is, and it creates a synthetic protein by bacteria and yeast cells. So, this would be creating things like the human growth hormone or human insulin. Bacteria can produce this, and we can get that synthetic protein that can then go help people who struggle in deficiencies with insulin, so people with type 1 diabetes. We can also see this create new vaccines and produce safer vaccines. So we can get subunit vaccines, which is a new approach to stimulate immunologic memory. This happens by introducing a gene of a pathogen into fruits and vegetables, or injecting humans with a plasmid carrying gene from the pathogen. Then our bodies go and take that pathogen and synthesize pathogen proteins so our bodies can identify these proteins and fight against them when we get infected with this bacterium. We can also do genetic screening using DNA microarrays, and they are used to screen individuals for inherited diseases caused by mutations. So things like trypsomies, so on and so forth. We can also look and identify pathogens in DNA in bloods and tissues. So we can take someone's DNA and identify if there's a bacterium or a pathogen present, because our body has broken down that DNA and our blood's used to remove it. DNA fingerprinting is identifying individuals or organisms by their unique DNA sequence. So we can take those restriction enzymes, cut them up, and insert or find genes that we need and run them through a gel electrophoresis. We can also use gene therapy, so genes can be added to missing or defective genes are replaced with normal copies. Sometimes we can see patients have immunological responses because your body is meant to recognize things that aren't self, and if you're changing the DNA, then your changing self. Medical diagnosis, so we can take patient specimen and examine them for presence of gene sequences unique to pathogens. And we can also use xenotransplants, where we take animal cells, tissues, or organs and introduce them into the human body for some kind of use. So tendons, heart valves are typically replaced by pigs, because there's not a lot of blood flow and your body won't um, reject them as easily. We can also see agricultural applications. So we can see the production of transgenic organisms, where recombinant plants and animals altered by the addition of genes from other organisms. So we can see things like herbicide tolerance, where genes are added so farmers can kill weeds without killing crops, right? So the gene from agrobacterium conveys resistance to Roundup. So if we put that gene in our corn plants, then we can kill the weeds around the corn and not kill our own food supply. We can also do salt tolerance. So we can insert genes for salt tolerance into tomatoes and canola plants, and these plants survive, produce fruit, and remove salt from the environment. So it's a win-win. We get food and they remove salt. We can also have freeze resistant. Crops can be sprayed with modified bacteria that can tolerate mild freezes, as well as things like pest resistance. So the Bt toxin, which is a naturally occurring toxin that is harmful for insects, can be used by organic farmers to reduce the insect damage to crops. We can insert the Bt toxin into various crop plants, and then when the bugs come and eat them, they die from that toxin. We can also see improvements into nutritional yield. So we saw this as well. So enzymes that break down pectin is suppressed in some tomatoes. This allows tomatoes to ripen on the vine and increase shelf life. We also have bovine growth hormone, which allows cattle to gain weight more rapidly, and they have a lower fat content, about 10% lower, and produce about 10% more milk. The gene for beta carotene, a vitamin A precursor, can also be inserted to rice, and we need this so we can ingest and take up vitamin A. Scientists considering transplanting gene codes for other metabolic pathways as well. All right, that is it for today. I will see y'all next time.